Naive B cells can only express surface immunoglobulin of the IgM and IgD isotypes. In these cells, the heavy chain immunoglobulin locus has previously undergone VDJ recombination that placed rearranged V, D, and J segments upstream of the C mu and C delta constant regions. Transcription initiated upstream of the rearranged VDJ segments can produce alternatively spliced mRNA transcripts that generate either IgM heavy chain or IgD heavy chain proteins. After translation, the functional IgM and IgD proteins associate with light chain proteins and become expressed on the cell surface. In this initial orientation, transcription of immunoglobulin arises only from the promoter located upstream of the rearranged VDJ and produces only IgM and IgD isotypes. The gene segments encoding the constant region for all of the remaining isotypes are located downstream of the C mu and C delta constant regions. In the mouse genome, these constant regions occur in the order of C gamma 3, C gamma 1, C gamma 2b, C gamma 2a, C epsilon, and C alpha. With the exception of C delta, each of these constant regions is preceded upstream by a sequence of DNA known as a switch region. Switch regions range in size from a few kilobases to nearly 10 kilobases in length and are composed of hundreds of repeats of G-rich short DNA sequences characteristic of these regions. For example, the switch region located upstream of the C mu constant region is approximately 3 kilobases in length and is composed of approximately 150 copies of this short 21 nucleotide element, which itself contains 3 copies of the pentameric sequence GAGCT. Located upstream of each switch region is a DNA sequence that functions as a promoter, called an I region promoter. Under appropriate circumstances, the I region promoter may be induced to initiate transcription by RNA polymerase II. Transcription begins just upstream of the switch region and continues through the region of DNA encoding the constant region. When this first occurs, the messenger RNA produced is known as a sterile transcript because it lacks an initiating methionine codon and therefore cannot be translated into functional protein by the ribosomal machinery. Isotype switching is stimulated in the course of an immune response. An activated T follicular helper cell engages a B cell through linked recognition. The follicular T cell expresses CD40 ligand and produces cytokines, in this example interferon gamma. CD40 ligand engages a CD40 molecule on B cells that activates NF-kappa B, and interferon gamma activates STAT1 within the B cell. These transcription factors cooperate to selectively induce the transcription of the I region promoter upstream of the C gamma 2A constant region. However, they do not induce transcription from other I region promoters, such as C epsilon or C alpha. The different transcription factors activated by the cytokines determine the specificity of I region activation. In this case, activation of STAT1 by interferon gamma leads to the activation of the I promoter region for C gamma 2A. Induction of transcription in the I region promoter opens up the DNA and allows an enzyme known as activation-induced cytidine deaminase, or AID, to access the switch regions. As RNA polymerase II transcribes the sterile transcript, the repetitive elements in the switch region induce a behavior in the polymerase that allows AID to deaminate single-stranded cytidine residues in the DNA and convert them randomly to uracils throughout the switch region. Through the action of 2 other enzymes, uracil DNA glycosylase, or UNG, and apyrimidinic endonuclease, or APE1, these uracils are eventually converted into single-stranded nicks in the DNA on both of its strands. These processes also occur at the C mu region or any other switch regions actively transcribed in the B cell. These activities create multiple breaks in the DNA of the mu switch region, as well as the DNA of the switch region selectively activated by cytokines. Because the breaks have been produced in two distinct locations at a great distance from one another, the double-strand break repair machinery joins the two switch regions and excises all of the DNA located between the two damaged regions. After the double-stranded DNA break repair machinery finishes its work, the damaged switch regions will have been spliced together at some random point, leaving remnants of each switch region on either side of the repair joint. The rearranged VDJ segment is now placed a few kilobases upstream of a new constant region, in this case C-gamma 2A. This now makes it possible for transcription initiated from the V region promoter to generate a transcript encoding the original variable region, but now in the context of a different constant region. When the messenger RNA is fully spliced, the intervening switch region sequences are removed, leaving only the segments encoding a fully functional C-gamma 2A heavy chain. The B cell that has undergone isotype switching will begin to secrete antibody of the new isotype class.